DNA stands for deoxyribonucleic acid. It has 4 bases: adenine, thymine, guanine, and cytosine. So DNA has 2 strands, or it is 2-stranded, and it carries genetic information, and therefore it enables transfer of genetic information from the parent to the offspring, and also the information is unchanged. So DNA replication. So during the DNA replication, the double helix unwinds and the strands of the DNA, the two of them, they unzip themselves due to the weak hydrogen bonds that link the bases. So that information is copied onto the new DNA structure using a parent template. The parent DNA replicates itself, and then the replication makes it possible to pass the DNA with the exact genetic information to the daughter cells. So as the cell prepares for cell division, during interphase, DNA normally replicates itself, or it doubles itself. DNA is important in protein synthesis. Therefore, it makes the structural proteins that determine the structure of an organism, the functional one that determine how the organism functions, for example, enzymes that speed up the rate of biological reactions, hormones that are used for communication in the body. So the sequence of bases along the DNA acts as a code which determines the sequence of amino acids. So the code normally has 3 bases that follow each other, forming a codon, and normally it codes for production of a particular protein. For example, AAA, that one codes for lysine amino acid, TTT for phenylalanine, CAA for glutamine, GTT for valine, and CTA for leucine. So protein synthesis takes place in the ribosomes, in the cytoplasm, and the RNA acts as a link. It links the DNA. It is the messenger RNA communicates between the nucleus and the cytoplasm of the cell. So messenger RNA, it is formed from DNA strands, and appropriate section of DNA acts as a template that is copied. So the double helix then unzips, and then the nucleotides align themselves along the template of the base sequence that is there. Then the base sequence is copied onto a new strand, which becomes RNA. So for the RNA, thymine is replaced with uracil, so there is no thymine in RNA. So after that, messenger RNA leaves the nucleus with full information or instructions about the kind of proteins that is needed, and then it goes into the ribosomes with that information, and it is translated by the ribosome, and then it is used to assemble the amino acids that are needed. So that assembling is done by a transfer RNA. So transfer RNA transport amino acids from the pool into the cytoplasm to the protein assembly point, depending on the sequence of the messenger RNA. Amino acids are condensed to form long polypeptide chains, and therefore genetic information is stored there. So we're then going to look at the first law of heredity that was by the Grigol Mendel. He determined the height of the stem, tall and dwarf, height of the texture of the seed coat, smooth or wrinkled, color of the flowers, either purple or white, position of the flowers, either axial or terminal, and the color of the cotyledon. So the dwarf plants self-pollinated to give rise to dwarf purebred, while the tall were self-pollinated to give rise to tall purebred. So when purebred dwarf plants were crossed with purebred tall plant, then he got a mixture of both tall and dwarf. When the purebred garden pea were crossed with the purebred variety, all the offsprings were tall, and that was named as the first video generation. When the tall offspring were crossed from the first video generation and planted, The second fetal generation had a mixture of tall and dwarf in the ratio of 3:1. So the role of segregation and then the role of independent assortment, the characteristics of an organism. In short, they are determined by the factors which occurs in pair. That is how Gregor Mendel was able to conclude. Reasons for Mendel's success. 1. He used favorable materials. The pea plants were readily available. They were self-fertilized. And then the pea plant he used had several observable traits, like the texture, the height, the color of flowers, so he could be able to test using different traits. And then his study was focused on a particular traits, while those from before him had been attempting to determine the wholesome heredity. And then he kept data, accurate data of all his experiments for analysis. Monohybrid inheritance characteristics, for example, height, it is controlled by a single pair of genes that is controlled by a single pair of hereditary factors. Then it involves transmission of a pair of traits from e.g. tallness and dwarfness, smooth and wrinkled coats, and then the genes occurs in pairs. So those pairs of genes are called alleles, or we say that alleles are alternative pairs of genes. So the genotype is the genetic makeup of an organism. Phenotype, they are the observable features, how the organism appears from outside. So in his experiment, the capital letter represents the dominant gene, that is the gene that is expressed wherever it's present. And then the small letter represents the recessive gene. It's a gene that is expressed in absence of a dominant gene. The symbol for male and female are as represented. So when writing the genetic diagram, you start with parental phenotype. You start with male or female. Parental genotype, now that is its genetic makeup. Dwarf is represented by small letters. It's a recessive trait. Small t, So then you find the gametes, you separate them as I have done there, and the gametes are always circled. So you circle the gamete individually and then you cross the first one here to the first one on the other side. Make sure that your crosses are well visible and clear. So you can see the first fetal generation, all the plants that resulted were dwarf. He crossed purebred dwarf with dwarf plants, and therefore it resulted to dwarf plants. So when he crossed the tall plants with the tall plants, of course it resulted to all tall plant. That is parental. You start with the parental phenotype, male and female, tall, genotype like that. Gametes, you separate them and you circle, and then you write the phenotype. All of them were tall. When the tall were crossed with the dwarf, then it resulted into a mixture of tall and dwarf. So parental phenotype, there you have a tall plant and a dwarf plant. Gamete, you have capitals and capital T and small t. Then when you cross them, you find one is tall. The second one is also tall because capital T is dominant over small t. The third one is tall, and the last one is also tall. So if the offspring was self-pollinated they produce offsprings that are tall and dwarf. That is, if he took those offspring that had a mixture of tallness and dwarfness, then he crossed them. As you can see, all of them were tall, but one of them was dwarf. So a plant can be tall, but it possesses the gene for dwarfness. That is how that experiment is all about. Complete dominance, as I have said earlier, our dominant gene is completely dominant over the recessive gene. It completely masks the expression of a recessive gene in heterozygous condition. Heterozygous condition is where both of them are present. So for homozygous allelic genes are identical, that is complete dominance. They can be either capital T or small t. Those are homozygote. Homo means the same. Heterozygous, it's where the allelic are not identical individuals, is known as a heterozygote. It could be a mixture of tallness and dwarfness. So here, in tallness and dwarfness, capital T is dominant. It expresses itself in homozygous condition and also heterozygous condition. Small t is recessive. It expresses itself only in homozygous state. So you find that these monohybrid crosses are based on Mendel's first row characteristics of organisms and determined by internal factors. So these internal factors are the genes. When you look at the probability, the chances were 3 tall and 1 dwarf. So you write it in form of either fraction or ratio. It could be 3/4 are tall, 3/4 were dwarf. So in heterozygous, the chance of producing capital T gamete from both parents were half. It was a chance. When you're separating these gametes, the chance of producing capital T is half. The chance of producing small t is half. That is why when you separate the gametes, we say that the probability was a quarter. The probability of you having capital T is a quarter, capital T small t is a quarter, capital T small t is a quarter, and also small t is a quarter. Then the other one was using Drosophila melanogaster. So they used Drosophila. These scientists used Drosophila because of its wing length. Its wing length trait was dominant over vestigial wing. Its eye color, the red eye color, was dominant over the white. Size of the abdomen, broad abdomen, was dominant over narrow. Body color, gray, was dominant over black color. So he used Drosophila as a favorable organism. So the reason as to why Drosophila was very suitable is that the female raised many eggs, resulting to many offsprings at once. So therefore there is a high sample size or large sample size. They are easily bred in the lab. They have a short generation time. The offspring can be crossed with their parents, and then the flies are safe to handle. They do not have any diseases. In complete dominance, it's shown in 4-o'clock plant, where a cross between a red flowered plant and a white flowered plant results in F1 generation with all offsprings having pink flowers. That means that the red color is not completely dominant over the white, neither is the white completely dominant over the red. So in this case, both the colors are expressed, so that is incomplete dominance. So that is also another example. Red and white, if they are crossed, they will the F1 generation, if they were crossed, red and white, they will give pink offspring. The inheritance of ABO, it is transmitted from parent to offspring, of course. As we said earlier, the blood group is also determined by your parent. So it has 3 genes involved. There is alleles that are responsible for presence of this antigen type on red blood cell. There is gene A responsible for presence of A, gene B responsible for B, O responsible for no antigen on the red blood cells. So all of them, they are called dominant. That means AB will be expressed together, and O is a recessive gene, so it is only expressed in absence of the other two, in absence of AB. So a person with genotype blood group A has genotype AO or AA with antigen A. Blood group B has BB or BO. BO in the sense that O is recessive. And then AB has AB having both antigen A and B, and then antigen blood group O, they do not have any antigen. So a man with blood group A and a woman with blood group B produce heterozygotes. The phenotype is male and female. Blood group, the parental genotype, because they are producing a child with AB, then they have to be heterozygote. So AO having A and O and BO, when you cross them, you get blood group AB, A, B, and O. So the fact that there is O then means that those people were heterozygote. If a man is homozygous for blood group A, married a woman who is homozygous for blood group B, they would only produce children with AB. So phenotype there is male and female. Parental genotype AA, BB, AABB. When you separate the gametes and then when you cross them over, you find A will be with B, AB again, AB, and then AB. So all of them will have blood group AB. Rhesus factor. People who are rhesus positive, they have rhesus factors. Those with rhesus negative lack rhesus antigen. So that antigen is antigen D, and it is controlled by presence of the rhesus gene. You find that in your blood group, some people are O positive, others are O negative. So if you are positive, it means you have this rhesus antigen. The presence of that rhesus gene is dominant over its absence. So the rhesus positive individual can be homozygous or heterozygous. That means they can be Rh positive, or Rh positive, Rh negative. But the rhesus negative people can only be homozygous for that trait. So those two are expressed together, like you can see there, rhesus positive, rhesus negative, or rhesus positive, rhesus positive. So the gametes there, when you separate them, it will be rhesus positive, rhesus negative, rhesus positive, rhesus negative. And then when you cross them, you find that rhesus positive, rhesus positive, rhesus positive, rhesus negative, and rhesus positive, rhesus negative. So you have 3 of them having rhesus positive and then 1 rhesus negative. Then if both parents of the children are rhesus negative, so rhesus negative, you just express it like that, then you find that the children will be rhesus negative. Determining unknown genotypes. So if the garden pea gene was determined red flower is dominant over that determine white flowers, a plant with red flower may either be homozygous or heterozygous. So genotype is to establish the genotype, you cross that plant with a homozygous recessive plant. So if the offspring will have a mixture, then it will show that it was not homozygous. But if the offspring will only bear the red flowers, it shows that the plant was homozygous. So that is how you carry it out. Parental genotype, parental phenotype first, parental genotype. When you separate the gametes, you have capitals and then small, and then you cross them, you find all are red. Remember, capital R is dominant over small r. So if the offspring bear a mixture of white and red color in equal proportion, then the red flowered plant was heterozygous, like this one of us. It is red flowered plant and white flowered plant, they were crossed. Then you have the parental genotype, parental phenotype there. So there are 2 red and 2 white. So there you're supposed to have small r, not capital R. Here it's small r for white. So then you'll have 2 red and 2 white. So a cross between individuals showing dominant trait with another individual showing recessive trait. That's what it's called a test cross. and it is used to determine the unknown genotype. Back cross, on the other hand, it's a cross between offspring and one of its parent. So phenotype, if the parent is phenotypically tall, they could be homozygous or heterozygous, as shown here. Homozygous or heterozygous. Heterozygous is like this one. You have capital and small t. Yeah, homozygous tall. Heterozygous is when both of them are the same, like that. Sex determination. So sex is also determined by a pair of chromosomes, and humans have 3 pairs of homologous chromosomes. They have 23 pairs of homologous chromosomes. That means in total they have 46 chromosomes. So the genes that determine whether a child is male or female, they are located on a specific pair of chromosomes that are known as sex chromosome. That is normally chromosome 23. So a male has XY, so he is heterogametic, where a female has XX. They are homogametic. Those are the chromosomes, the sex chromosomes of a male and a female. So after meiosis, the male sperm can either have X or Y. So the sex of the child is a matter of chance also. It depends on whether the sperm that will fertilize the egg has X chromosome or it has Y chromosome. If the sperm has X chromosome, of course the female also has an X, so they will be the girl will be produced XX. If the chromosome of the male carries Y, then the woman also has X. They will get a baby boy. So it is by chance that a baby born is a boy or a girl. So for example, here you have parental phenotype, male and female parental genotype. That is XY. You've said the male is heterogametic, woman is XX, and the offspring genotype will be XX, XX. Those are X, the girls, and then the boys, they have XY. So these people can have either boy or a girl. So there are 2 in 4 chances that the baby's born will be, or the baby that is born is a girl. There are also 2 in 4 chances that the baby born is a boy. So probability here is half. So in short, the probability or the chance that a boy or a girl is produced in the family is a half. The baby can be a boy or a girl depending on the chance. So in other animals, the male is homogametic while the female is heterogametic. For example, in birds, the male is XX while the female is XY. In insects, the female is XX while the males are XO. So those are some genotypes that you need to notice. The Y chromosome is missing. And also when you are told to do cross for sex determination in Drosophila, it is just like that of a man, the male or human. That is, male is X, Y, female is XX. Sex linkage. Here, genes located on the same chromosome, they are known as sex-linked genes, and they are located on the sex chromosome, and they linked genes do not segregate. Genes located on the same chromosome, they are known as sex-linked genes, linked genes, sorry, and they do not segregate during meiosis. They just remain the same.